Namaskar. Welcome to the subject Olfaction, and today's session deals with olfaction in human system. Firstly, coming to the introduction. Olfaction, known as the sense of smell, is a very important property for evaluation of food products, either for acceptance or rejection, either for liking or disliking, as well as for imitation flavored products development. Though it rates next to taste in its depthness of study, but the food cannot be accepted upon if the smell is not desirable. The sense of smell is mediated by specialized sensory cells of the nasal cavity. The odor responsible compounds binds to specific sites of olfactory receptors, and transmission of signal to the brain reveal the type of odor, its liking, its comparison to the concept, etc. Olfaction, along with the taste, is a form of chemoreception. The chemicals themselves activate the olfactory system and can be sensed at very low concentrations. Further, as a result of stimulation by chemical compounds in the respiratory tract, leads to specificity in odor recognition. Smell forms as one of the sensory sensations of flavor besides the taste and tactile properties. The present topic will be dealt under the headings of definition of odor and importance, anatomy and physiology of olfaction, and the mechanism of odor perception. Odor can be defined phenomenologically through the nasal cavity action as well as physiologically through the response of the nasal cavity action. Odor is the property of a substance or substances that is perceived in the human and higher vertebrates by inhalation in the nasal or oral cavity, that makes an impression upon the olfactory area of the body which is distinct from hearing, tasting or feeling. It does not cause as a result of choking, cooling, warm, drying, wetting, which are the attributes involved by other sensory organ. On the other hand, physiologically, odor is the sensation perceived from the responses of the olfactory nerve or first cranial nerve. The three elements of odor are intensity, type and variety. The type of odor refers to the source, say fruity, spicy, aromatic odor, etc., and the intensity speaks of the depthness of perception or the concentration. While the variety makes a difference, may be a mixture of odors, may be the unripened, ripened or senescence of particular fruit odor, sweetish, acidic or stages of foul odors. Ultimately, the variety has a greater impact on liking or disliking or rejection decisions. The importance of odors in food industry refers to the acceptance of the various food products, since odor can attract or repel consumers. The original or normal odor of the ingredients used or sources used add to the positive strength of the product, but the processing effect, byproducts wastage emanated of odors leads to environmental pollution and health hazards. At times, modification of odors is attempted to overcome or mask the undesirable odor character, thereby malodor's intensity is reduced. Some of the masking agents such as vanillin, citral compounds mask the odd odor with their powerful strength. The sensory combination and integration of odors, taste, oral irritations, thermal sensations and mouthfeel that arise from a particular food is referred to flavor. Olfaction, taste and trigeminal receptors together contribute to flavor. The human tongue can distinguish only among 5 distinct qualities of taste, while the nose can distinguish among hundreds of substances even in minute quantities. During inhalation phase, smell is perceived, while during exhalation phase, olfaction contributes to flavor. The olfactory system is the only human sense that bypasses the thalamus and connects directly to the forebrain, which is called frontal lobe. Now next is the anatomy and physiology of olfaction. In humans, the olfaction process occurs through the sensory organ nose. The two nasal cavities are separated by a smooth median septum. The lateral walls of the cavities have a series of folds approximately horizontal, varying from 2 to 6. The lower fold extends over most of the length of the nasal cavity. The two folds above this, called conchae, are smaller and protrude into the cavities. It provides three channels called the inferior, median and superior meatus. Figure 1 shows you the cross section of the nose. Each meatus is confluent with the large common meatus, which communicates with the olfactory cleft above the superior concha and the septum. These spaces are all connected by very narrow passages with the posterior nares and with the pharynx. The upper and lower nasal passages are mere slits, nowhere wider than 1 to 2 mm. In normal inspiration, the fraction of inspired air passing the olfactory slit is 5 to 10%. For small rates of flow, the number of molecules striking the olfactory region is small because so many are being absorbed by the mucous membranes. At higher rates of flow also, the number of molecules striking the olfactory region is small, since small part of molecules in the olfactory slit can diffuse to the walls. Therefore, about 2% of odorous molecules reach the olfactory epithelium in normal breath. With the injection or by blast techniques, it may rise up to 4%. Olfaction is anatomically different from gustation because it uses the sensory organ nose and nasal cavity. Olfactory sensitivity is directly proportional to special area in the nose, specifically the olfactory epithelium. Humans have about 1.6 square inches of olfactory epithelium. The area in the nasal cavity near the septum is reserved for the olfactory mucous membrane, where olfactory receptor cells are located. In humans, there are about 10 million olfactory cells, each of which has 350 different receptor types composing the mucous membrane. Each of these 350 receptors types is characteristic of only one type of odorant. The deeper part of the nasal cavity contains a pseudo-stratified columnar epithelium which contains ciliated cells. These cells are hair-like projections that contain olfactory receptor proteins. These proteins carry out the transduction of odorantza into electrical signals for neural processing. The cavity also contains many alveoli tubular glands with mucous cells. During normal breathing, air does not reach easily the olfactory cleft, and upon expiration, air normally passes along the lower roots. Therefore, direction of projection of odoriferous molecules by sniffing draws up the air up into the olfactory cleft. This point is of concern in finding out the odor of the products and measurement techniques followed for odor evaluation. However, the air reaching the olfactory cleft is influenced by the nasal obstruction, shape of the nose and the condition of mucosa. When mucosa is pale and dry, the detection of smell will be weaker. In addition to the olfactory neurons, epithelium contains basal cells, supporting cells and sensory, that is olfactory cells. It is estimated that man has 10 to 20 million such receptors. Each olfactory receptor cell is a primary sensory bipolar neuron. The olfactory neurons are unique because they are generated throughout the life by the underlying basal cells. New receptor cells are generated approximately every 30 to 60 days. The bipolar olfactory neurons have a short peripheral process and a long central process. The peripheral process extends to the mucosal surface to the end of the olfactory knob, which has dense ciliated cells at the mucosal surface. These cells express the olfactory receptors which interact with the odorants, which react with the odorants. The olfactory epithelium, as well as the rest of the nasal surface, is innervated by bare nerve fibers from the trigeminal nerve. The nuclei of the receptors are situated at different depths in the olfactory mucosa, with olfactory rods of 20 to 90 micron long. This distribution may be related to differential sensitivity to odors and odor quality discrimination. Next, let us look into the mechanism of odor perception. The process of odor perception starts from the sniffing of odorous material, which through air currents reaches the olfactory receptors, followed by binding. This, through cranial nerves, reaches olfactory bulb in the brain, then signal transmission reveals the odor. In a nutshell, the schematic representation of olfactory perception is outlined in figure 2. Here in figure 2 is the diagrammatic scheme of olfactory perception. Source: Principles of Sensory Evaluation of Foods, 1965, page number 163. The odorous material must be volatile and should reach the olfactory cleft, thereby the receptors. The odor must dissolve in an aqueous mucus and diffuse through it. The mucus overlying the epithelium contains mucopolysaccharides, salts, enzymes and antibodies. Thus, mucus acts as a solvent for odor molecules. It flows constantly and is replaced every 10 minutes. Olfactory sensory neurons project axons to the brain within the olfactory nerve. These nerve fibers, having myelin sheaths, pass to the olfactory bulb of the brain through perforation in the cribriform plate, which in turn projects olfactory cortex and other areas. The axons from the olfactory receptors converge in the outer layer of the olfactory bulb into small structures of less than 50 micrometer in diameter called glomeruli. Mitral cells located in the inner layer of the olfactory bulb form synapses with the axons of the sensory neurons within the glomeruli and send the information about the odor to other parts of the olfactory system, where multiple signals may be processed to form a synthesized olfactory perception. A large degree of convergence occurs with 25,000 axons synapsing on, say, 25 mitral cells, with each of these mitral cells projecting to multiple glomeruli. The mitral cells leave the olfactory bulb in the lateral olfactory tract. These synapses on 5 major regions of the cerebrum, that is the anterior olfactory nucleus, the olfactory tubercle, the amygdala, the piriform cortex and the entorhinal cortex. The piriform cortex projects to the medial dorsal nucleus of the thalamus, then projects to the orbitofrontal cortex. The orbitofrontal cortex mediates conscious perception of the odor. The 3-layered piriform cortex projects to a number of thalamic and hypothalamic nuclei, the hippocampus, amygdala and the orbitofrontal cortex. But its function is not well understood. The entorhinal cortex projects to the amygdala and is involved in emotional and autonomic responses to odor. It also projects to the hippocampus and is involved in motivation and memory. This is possibly due to the olfactory system's closed anatomical ties to the limbic system and hippocampus areas of the brain that have long been known to be involved in emotion and memory respectively. Since one receptor is responsive to various odorants and there is a great convergence at the olfactory bulb, it looks strange that human beings are able to distinguish so many different odors. Probably, there must be a highly complex form of processing wherein many neurons in the olfactory bulb are responsive to many different odors. However, half the neurons in the orbitofrontal cortex are responsive to only one odor and the rest for a few. This process is influenced by the inhibition and excitation of mitral cells, neuromodulators such as acetylcholine, serotonin and norepinephrine. In fact, inputs from the two nostrils have separate inputs to the brain, with the result for humans to experience perceptual computation in the olfactory sense. Further, the perceived odors undergo coding in brain prior to the signal to give a final decision of the odor experience. It is possible to find the spatial coding through which brain is able to distinguish specific odors as well as temporal coding, which is important. Thus, the process of coding is of great concern, however, still being researched. When the odorant is detected by receptors, the receptors break the odorant down and then the brain puts the odorant back together for identification and perception. The odorant binds to receptors which recognize only a specific functional group or feature of the odorant. Thus, the chemical nature of the odorant plays the role in perception. After binding the odorant, the receptor is activated and will send a signal to glomeruli. Each glomerulus receives signals from multiple receptors and detects similar odorant features. Because multiple receptor types are activated due to the different chemical features of the odorant, multiple glomeruli will be activated as well. All the signals from glomeruli will be sent to the brain, where the combination of glomeruli activation will encode the different chemical features of the odorant. Then, the brain will put the pieces of the activation pattern back together in order to identify the odorant. The recent research has suggested that the average individual is capable of distinguishing between over 1 trillion unique odors. The poor performer could still distinguish 80 million odors. The identification of different odors, also influenced by the genetic differences and a couple of genes, has been linked conclusively to particular smells. For example, the receptor OR5A1 and its genetic variants are responsible for the ability to smell beta-ionone, a key aroma compound in foods and beverages. The odorant receptor OR2J3 is associated with the ability to detect the grassy smelling odor. Cis-3-hexane-1-ol, the receptor OR6A2, is linked to coriander odor. In humans, the sense of olfaction is stronger in females than the males. In females, the sense of olfaction is strongest around the time of ovulation and during the phase of menstrual cycle. Humans can also detect individuals that are blood-related kins from olfaction. Mothers can identify by body odor their biological children. Functional imaging shows that this olfactory kinship detection process involves the frontal temporal junction, the insula and the dorsomedial prefrontal cortex, but not the primary or secondary olfactory corticals or the related piriform cortex or orbitofrontal cortex. As the age advances, the number of olfactory neurons steadily decreases, thereby the sense of smell gets reduced. Loss of smell has been linked to inadequate nutritional intake, reduced social pleasure and decreased psychological well-being. The abnormalities of olfaction also play a role in the process of perception of odor. The dysfunction can result from pathologic processes at any level along the olfactory pathway. It may be of two types, that is conductive defects and sensory neural defects. In conductive defects, transmission of an odorant stimulus to the olfactory neuroepithelium is disrupted, while sensory neural defects involve more the central neural structures. The most common causes of primary olfactory defects are aging, narrow nasal passage, sinus problem, upper respiratory tract viral infections, and head trauma. Inflammatory processes cause a large portion of olfactory defects. These may include rhinosinusitis, allergies, cold, cough, etc. The central neural involved infection diseases include viral infections, sarcoidosis, mucosal edema, and polyp formation. Endocrine disturbances of hypothyroidism, hypoadrenalism, diabetes mellitus may affect olfactory function. The toxicity of inhaled or systemic drugs can contribute to olfactory dysfunction. Thus, the olfactory anomalies caused are summarized as per the medical terms. Cryptosomia nasal passages, obstruction in nasal passages. Anosmia, temporary or permanent loss of smelling capacity. Hyperosmia, an abnormally acute sense of smell. Hyposmia, decreased ability to smell. Merosmia, loss of only certain odors. Heterosmia, false odors perceived. Autosmia, odor sensation in the absence of odor stimuli. Cacosmia, persistent perception of unpleasant odors. Phantosmia, hallucinated smell often unpleasant in nature. Olfactory reference syndrome, psychological disorder which causes the patient to imagine he or she has a strong body odor. In conclusion, olfaction being an important phenomenon followed by every being throughout the life has a prominent place for decisions of liking, accepting, or otherwise the foods eaten. The sense of smell is experienced by the odorous substances through the process of transduction. Odor is the sensation perceived from the responses of the olfactory nerve. Intensity of odor, type and variety are the important elements of odor. The olfaction process occurs through the sensory organ nose. The process of odor perception starts from the sniffing of odorous material which through air currents reaches the olfactory receptors followed by binding. This through cranial nerves reaches olfactory bulb in the brain. Then signal transmission reveal the odor. The odorous material must be volatile and should reach the olfactory cleft, thereby receptors. The odor must dissolve in an aqueous mucus and diffuse through it. The mucus overlying the epithelium contains mucopolysaccharides, salts, enzymes and antibodies. Thus, mucus acts as a solvent for odor molecules. The average individual is capable of distinguishing between over 1 trillion unique odors. The poor performer could still distinguish 80 million odors. The identification of different odors also influenced by the genetic differences, and a couple of genes has been linked conclusively to particular smells. The olfaction, taste and tactile attributes together contributes to flavor. During the sniffing of odors, the inhalation process smell is recognized, while during the exhalation process flavor is recognized. With the aging, the number of olfactory neurons steadily decreases, thereby the sense of smell gets reduced. The lifestyle, food habits, diseases, drugs usage generally lead to dysfunction of olfactory process, reflecting the disorder of smell. This refers to reduced perception of smell, loss of smelling capacity, sense of unpleasant odors, etc. The loss of smell has been linked to inadequate nutritional intake, reduced social pleasure, and decreased psychological well-being.